professor, physician, epidemiologist. I am Dr. Sri Banerjee.
Hello everyone, I'm Dr. Sri Banerjee, core faculty for the College of Health Sciences and Public Policy at Walden University, with a joint faculty appointment at Ross University School of Medicine. I would like to talk some about, today a little bit about just big data and the role of big data within not only the area of medicine, but also talking about how this approach can combine various types of data that is available. So let's go into some of the omics, and this was something, and I say this frequently. This is something that I was introduced to a number of years ago because of this conference that I attended in Philly, where it was an international conference, and there were a lot of medical doctors and also experts in the field of omics. So at the time, I only knew about one type of omics, probably metabolomics, but then I, of course, learned about genomics pretty early on. GWAS studies, they're not new. Genome-wide association studies, they've been around for a long time. The types of studies that are new are epigenomics. That is kind of new. What was, by 2013, all of the genes within the human DNA were cracked. So we now know all the genes which are present in the human body, but what is not known is the interaction with the environment, which happens after transcription. So post-transcriptional and post-translational modifications in the DNA allows for actually interactions to be passed from generation to generation. So looking at this, there's genomics. Genomic studies whole genomes of organisms that includes the complete set of DNA and genes. It is a multidisciplinary branch that focuses on the ID of genetic variants associated with disease differential drugs effect, transplant outcome, etc. So the GWAS approach allows the characterization of genetic variants related to complex diseases in human populations. So in the realm of transplantation, GWAS permits to highlight interethnic and inter-individual genetic variations. Then going into transcriptomics, so once the gene has been transcribed into mRNA, and transcriptomics, so any transcript, messenger RNA or nuclear RNA, produced during the process of transcription by a genome under specific circumstances or in a specific cell. It evaluates both quantitatively and qualitatively differential RNA rate to have better insight into disease mechanisms. So transcriptomics actually goes into quantitative PCR, DNA microarrays that estimate predetermined sequences, and RNA seq using high throughput sequencing to capture all transcripts. So there's non-coding, as I said earlier, I said nuclear, but it's actually non-coding RNA, and so you can study, there's techniques to study the non-coding portions as well, because what was interesting to understand is even those non-coding portions are actually playing an essential role in many types of biological processes, diseases, and potential biomarkers as therapeutic targets. So transcriptomics analysis has enabled us to grasp the impact of gene expression depending on the organism and its consequences on human disease outcomes. Then you have proteomics, which then is after translation. Global whey proteins quantification, location modification, interaction, and functions in genome scale. And then metabolomics is looking at metabolites and small molecules in the metabolome under a given condition. So this could be a result of genetic or environmental factors and provides a metabolic profile of the cell tissue. And of course, abnormal levels of metabolism. This is indicative of disease. So thinking about metabolomics and thinking about one that I've talked about a lot is C-reactive protein, which provides a kind of understanding of how there's mechanisms. But there's others that provide wonderful understanding of what is going on within the body. And one I'll bring up is bilirubin. That is one that sometimes, in fact, when I was born as well, I had a slightly higher level of bilirubin, which is actually normal in certain circumstances, for instance, in Gilbert syndrome. And so this is an interesting medical condition to look at. Anyway, so bilirubin is a good example. And by the way, bilirubin gets converted into stercobilinogen to then give feces the brown color, and then urobilinogen to give urine the yellow color. So a lot of these metabolisms, and of course drug metabolisms are going within either the liver or the kidneys. So kidney function, even a measure of GFR, I would argue that creatinine is unquestionably one of the cornerstone sort of metabolic factors. So thinking about now, I consider genomics, transcriptomics, and proteomics not to this level, but it has been around for at least 2 decades. But when we are starting to get into metabolomics, I think the understanding of this, definitely epigenomics and phenomics, these have developed more so in recent years. And I'll explain to you a little bit more about these. So metabolomics we just went over. Epigenomics is really interesting, and I explained to this in multiple ways, is that the environment is actually going back and creating DNA-associated proteins, which actually then goes back and induces mutational changes in the actual DNA sequence. Now, if these mutations are taking place in the somatic cells, then it doesn't get passed down to your offspring. However, if these mutations are going on within the germ cell line where meiosis takes place, then this gets passed on to offspring, and then understanding that how this is getting passed from the environment, which completely goes beyond what we used to think with the central dogma, because the understanding always was that genes, they stay the same, they don't change, but this is saying that the expression of genes in the environment actually going back and changing the genes. So this is a field that's still evolving and hasn't come to its final stage yet. The key role of epigenetic signatures, and this is an interesting concept, on biological processes, disease development, and other pathophysiologic states. By the way, in the transplantation field, a better understanding of epigenetic modifications of both donors and recipients may lead to biomarkers, improvement in transplant recipient selection and progress and overall prediction outcomes. I've actually chaired a dissertation where there was transplantation scoring systems and outcomes. And one important, now this is at a basic principle I'm not going to go into detail about, but one important interaction that takes place when you have transplantation is that the host actually rejects the transplant due to a sort of an allergic reaction. And then you have phenomics, which is, again, an emerging field and has full range of physical and biochemical traits belonging to a given organism, the phenome. So phenomics focuses on qualitative measures of traits and also concerns the characterization of genotype-phenotype associations. So the phenomics capabilities requires expertise from multiple fields, biology, data sciences, engineering, and this is, again, a very new and definitely evolving field, but it is a necessary complement to genomics, offers useful insights into important outcomes such as disease, and allows a better understanding of the genotype-phenotype map. So these are concepts that are very important in understanding omics and the relationship between omics and big data. Without understanding omics first, you cannot understand big data completely. You have to understand the underlying concept of omics. Thank you for listening.